But it folds. It just folds itself. Nobody tells it what to do. It reads the amino acid sequence and builds the correct shape from nothing.
And when it folds wrong, it converts every other copy of itself into the wrong shape too. Indefinitely, with no DNA involved. I have been sitting with this fact for several years, and I have not fully recovered.
A self-assembling nanoscale precision instrument with programmable therapeutic properties. I'm already drafting the Series B deck.
It is mildly concerning that the most enthusiastic person in the room also has unsupervised access to laboratory reagents.
I once left a sweater in the dryer too long.
Stop. We are going to cover this properly and correctly.
Dr. Silly, I need you to explain all of this immediately.
That is what this podcast does. Welcome to the Krusty Lab. Today we are covering protein structure, primary, secondary, tertiary, quaternary, and what happens when the folding process fails. It is one of the most consequential topics in cell biology and medicine. And yes, the end of the episode gets dark.
The end of the episode is where I live.
Subscribe now. I have been reliably informed that proteins are the molecular machinery behind all disease and all therapy. And learning how they fold is basically buying intellectual equity in 20 different drug pipelines simultaneously. This podcast is free. The compounding knowledge is not. Subscribe.
A protein is a polypeptide chain, a linear sequence of amino acids linked by covalent peptide bonds that folds into a specific three-dimensional shape. That shape determines its function. Proteins are enzymes, structural components, motors, transporters, receptors, signaling molecules, antibodies. They execute nearly every process in the cell.
Everything?
Almost everything. And the critical word is shape. A protein is only functional when it has the correct shape. The sequence encodes the shape. The shape determines the function. This is the central logic of protein biology.
Change the shape by one wrong fold, and you can go from a functional enzyme to a toxic aggregate to something that rewrites the folding of everything around it. The margin between works perfectly and destroys a brain is a few angstroms.
It is mildly sobering that the entire structural discipline exists to understand a margin measured in 10 billionths of a meter.
There are 20 canonical amino acids. Each has a central alpha carbon bonded to an amino group, a carboxyl group, a hydrogen atom, and a variable side chain, the R group. The R group defines each amino acid's chemical identity, whether it is hydrophobic, hydrophilic, charged, polar, or aromatic. The sequence of R groups along the chain is what drives folding.
20 building blocks, and from 20 building blocks you get the whole proteome?
The human proteome contains roughly 20,000 protein-coding genes, and post-translational modifications expand the functional diversity to hundreds of thousands of distinct forms, all from 20 amino acids combined in varying sequences and lengths.
I need a second. I've just been walking around with hundreds of thousands of different precision machines running inside every cell for my entire life, and I found out today.
20 inputs, infinite product configurations. This is literally the SaaS business model. I am overcome.
Amino acids are joined by peptide bonds. The amino group of one amino acid reacts with the carboxyl group of the next, releasing a water molecule, a condensation reaction, and forming a covalent bond between the nitrogen and the carbonyl carbon. This is the peptide bond. The resulting chain has a free amino terminus at one end and a free carboxyl terminus at the other. This directionality matters. The ribosome synthesizes polypeptides strictly from N-terminus to C-terminus.
The chain has a reading direction, and it cannot be reversed. The ribosome is very opinionated about this. If you try to run translation backwards, you get nothing. I've thought about forcing it. You can't.
The sequence of amino acids in the chain from N to C-terminus is the primary structure. This is level 1.
So primary structure is just the order?
The order is everything. A single amino acid substitution in the primary sequence can completely destroy function or create a new pathology. The canonical example is sickle cell disease. A single glutamic acid at position 6 of beta-globin is replaced by valine. One substitution. Valine is hydrophobic, glutamic acid is charged. Under low oxygen conditions, this single change causes hemoglobin tetramers to polymerize, deforming red blood cells into a sickle shape that occludes capillaries.
1. Amino acid.
One amino acid out of 147 in the beta-globin chain causes an entire spectrum of clinical pathology.
And sickle cell trait, one mutant copy, one normal copy, actually confers partial protection against malaria in endemic regions. Natural selection maintained a lethal mutation at population scale because the heterozygote has a survival advantage. Evolution doesn't care about fairness, it cares about allele frequency.
One base change with a protective heterozygous phenotype and a severe homozygous pathology. The risk-reward profile here is genuinely interesting from a population genetics standpoint. I'm not saying I want to invest in it. I'm saying I understand why nature did.
It is mildly troubling that natural selection and a venture portfolio are being evaluated with the same analytical framework.
Moving to secondary structure. After primary sequence, the chain begins to form local folded patterns, regular repeating structures within the polypeptide backbone. This is secondary structure. The two dominant forms are alpha helices and beta sheets. Both are stabilized by hydrogen bonds. And, this is critical, between the backbone atoms themselves, not the side chains. The amide nitrogen-hydrogen bonds to the carbonyl oxygen. In an alpha helix, each carbonyl oxygen bonds to the amide 4 residues ahead in the sequence, creating a right-handed helical coil with approximately 3.6 residues per turn.
So the backbone is hydrogen bonding with itself 4 spots ahead, like the chain is reaching forward and grabbing itself?
Precisely. It is a self-stabilizing structure. The helix forms because those hydrogen bond distances and angles are geometrically favored by the amino acids adopting the correct backbone torsion angles.
And alpha helices are mechanically interesting. They behave like coiled springs under axial tension. There's a coiled coil architecture in muscle proteins, myosin, keratin, tropomyosin, where multiple helices wrap around each other for tensile strength, a nanotube made from biological polymer. I've been exploring whether you can stretch one with optical tweezers.
Beta sheets form by hydrogen bonding between separate strands of polypeptide running alongside each other, parallel or anti-parallel, rather than within a single continuous stretch. The result is a flat or twisted pleated sheet. Beta sheets are common in structural proteins and in the cores of many globular proteins.
I've seen clouds that looked like that.
Like a beta sheet?
Like something that couldn't decide what shape to be.
It is mildly plausible that she is describing loop regions.
Squiven may be correct. Loop regions, also called random coils, though they are not actually random, connect alpha helices and beta sheets. They lack regular secondary structure, but have specific conformations essential to function, particularly at active sites. Tertiary structure is the full three-dimensional architecture of the protein. How every secondary structure element, every loop, every R group arranges itself in space to produce a single compact functional shape. This is where the protein becomes a specific molecular tool.
And what actually holds the 3D shape together?
Multiple forces working in concert. The dominant driving force is the hydrophobic effect. Nonpolar side chains avoid water. They are buried in the interior of the protein, forming a hydrophobic core. This is thermodynamically favorable. It increases the entropy of the surrounding water by preventing the ordered water shells that would form around exposed hydrophobic patches.
Hide the greasy parts inside, expose the functional surface to the environment. That is exactly how you structure a product launch. The messy infrastructure is internal. The clean interface is what users touch.
That is an unreasonably accurate analogy for the hydrophobic core.
I've inadvertently been describing protein folding for years.
It is mildly depressing that this realization brings him pride rather than pause.
Beyond the hydrophobic effect, hydrogen bonds between side chains, electrostatic interactions between oppositely charged residues, van der Waals contacts, and in many secreted proteins, covalent disulfide bonds between cysteine residues. Disulfide bonds form oxidatively in the endoplasmic reticulum and can lock the tertiary structure into a stable conformation.
DTT, dithiothritol, reduces disulfide bonds and collapses the structure. Take the DTT away. Let it reoxidize. It refolds. I've done this with several proteins. You can demolish and reconstruct a protein's architecture in a single afternoon. It's the best lab experiment that exists.
You can dissolve a protein's shape and it just rebuilds itself?
That is precisely what Anfinsen demonstrated in 1961. He denatured ribonuclease A completely, used urea to unfold it, and beta-mercaptoethanol to reduce all 4 disulfide bonds, fully disordered chain. Then he removed the denaturant. The protein spontaneously refolded to its native, fully active conformation.
The whole folding instruction manual is just in the sequence?
The sequence is the instruction manual. This is Anfinsen's dogma. The native state of a protein is the thermodynamic minimum energy conformation determined solely by its amino acid sequence under physiological conditions. No additional information is required.
With important caveats. There are proteins that genuinely cannot fold alone under cellular conditions. The crowded cytoplasm introduces kinetic traps that the test tube does not. But Anfinsen's core finding stands. Sequence encodes structure.
Which creates a paradox. Cyrus Levinthal calculated that if a 100 residue protein sampled every possible backbone conformation randomly, 1 per picosecond, it would take on the order of 10 to the 77th years to find the native state. The universe is approximately 14 billion years old.
But proteins fold in milliseconds.
They fold in microseconds to seconds. The resolution is the energy landscape, sometimes called the folding funnel. The protein doesn't search randomly. The free energy surface is shaped so that most partially folded states are energetically closer to the native state than to random conformations. The chain follows a gradient, many possible starting points, one downhill basin.
Nature solved a search problem with a complexity of 10 to the 77 by converting it into a gradient descent problem. That is genuinely elegant. Every machine learning algorithm published in the last 40 years is a reinvention of what proteins have been doing for 4 billion years. I find this simultaneously humbling and motivating.
Kelpie, proteins are doing gradient descent?
Gradient descent is doing proteins. Thermodynamics came first.
Even with a favorable energy landscape, the cell needs help. Chaperones are proteins whose function is to assist other proteins in folding correctly. As a new polypeptide emerges from the ribosome, its hydrophobic patches are exposed. In the crowded cytoplasm, where protein concentration is hundreds of milligrams per milliliter, exposed hydrophobics would immediately aggregate. Chaperones bind those patches, hold the chain, and give it shielded time to fold.
There are entire proteins whose whole job is to babysit other proteins while they figure out their shape?
The HSP70 family does exactly this. The chaperonins, GROL, GROES in bacteria, the Tri-C complex in eukaryotes, form a barrel structure that completely engulfs the unfolded protein and provides an isolated chamber for folding, a nanocage, an Anfinsen cage.
GROEL is 2 stacked rings of 7 subunits each, with a central cavity that changes shape on ATP hydrolysis, applying mechanical force to the substrate. It physically unfolds kinetically trapped proteins and gives them a fresh folding attempt. This machine is 900 kilodaltons, and it is doing active mechanical work on misfolded proteins using ATP. It is a molecular compressor that runs on chemical energy and outputs correctly folded proteins. I cannot overstate how beautiful this is.
A quality control service that physically reshapes defective units using ATP as fuel and releases conformationally compliant products. That is a manufacturing process. I want to license the architecture.
It is mildly characteristic of this group that a protein folding machine is being evaluated simultaneously as a drug target and a franchise model.
Moving to quaternary structure, many proteins do not function as single polypeptide chains. They assemble into complexes of 2 or more subunits, each with their own tertiary structure, and the multi-chain complex is the functional unit. This is quaternary structure. The interactions between subunits use the same forces as tertiary folding: hydrophobic contacts, hydrogen bonds, electrostatics.
So proteins can work in teams?
They can, and sometimes the team has properties that no individual member can produce alone. The canonical example is hemoglobin. Hemoglobin is a heterotetramer, 2 alpha-globin and 2 beta-globin subunits, each carrying a heme group that binds oxygen. 4 subunits, 4 binding sites.
And the binding is cooperative. When one subunit binds oxygen, it undergoes a conformational shift. The T state relaxes to the R state, and that shift propagates mechanically through the quaternary interface, increasing the oxygen affinity of the remaining subunits. The Hill coefficient for hemoglobin is approximately 2.7. These are not 4 independent binding events. They are communicating through quaternary structure.
The subunits talk to each other?
Through shape change. One binding event deforms the interface, the deformation shifts the partner becomes more receptive. Information is transmitted through mechanical coupling across the quaternary structure with 0 molecules of signaling protein involved.
Cooperative binding means hemoglobin loads oxygen efficiently in the lungs at high PO2 and releases it efficiently in the tissues at low PO2. Compare this to myoglobin, a single subunit oxygen storage protein with a hyperbolic binding curve and no cooperativity. Myoglobin binds oxygen tightly at all oxygen tensions. It cannot load and unload efficiently across a physiological gradient.
Quaternary structure enables emergent cooperative behavior that no individual subunit can produce. You know what else works that way? Networks. This is network effects applied to molecular biology. I am extremely moved.
My grandmother had a thermos.
What?
It held more at higher temperature, less at lower. The amount it held depended on where you were.
She is describing context-dependent binding behavior. That is, closer to the sigmoidal oxygen dissociation curve than I would like to admit.
It is mildly inevitable.
Now, misfolding. Protein misfolding occurs when a protein does not fold into or cannot maintain its correct native conformation. Hydrophobic patches that should be buried become exposed. In the crowded cellular environment, exposed hydrophobics aggregate. The aggregates can be toxic, and in the worst cases, the misfolded protein actively converts correctly folded copies into the misfolded form.
Toxic aggregates. Okay, what kind of diseases are we talking?
Alzheimer's disease, Parkinson's disease, Huntington's disease, type 2 diabetes, cataract formation, a whole family of prion diseases. Misfolding is not an edge case. It is a fundamental failure mode of protein biology that underlies some of the most prevalent and devastating conditions in medicine.
And the aggregates have a specific architecture. They're not random clumps. Misfolded proteins organize into amyloid fibrils, a cross-beta sheet structure, where beta strands run perpendicular to the fibril axis, hydrogen bonding along the length, extremely stable, insoluble, resistant to proteases. The cell cannot easily clear them.
The pathway from misfolded monomer to fibril follows a nucleation elongation mechanism. Misfolded monomers associate into small oligomers. Oligomers elongate into protofibrils and mature fibrils. And here's the counterintuitive finding from the last 2 decades of Alzheimer's research. The small soluble oligomers are the most cytotoxic species, not the large insoluble plaques.
The small ones are worse than the big ones?
The oligomers disrupt neuronal membranes, impair synaptic vesicle trafficking, trigger inflammatory cascades, and activate caspase-dependent apoptosis. The mature plaques are relatively inert end-stage deposits. They are a marker that the pathology has been running for years, possibly decades.
This is why 2 decades of Alzheimer's drug trials targeting amyloid plaques largely failed. The drugs cleared plaques. The patients did not improve. You were hosing down the ash after the fire was already out.
You targeted the endpoint marker instead of the active toxic species and spent billions on phase 3 trials. That is the most expensive misalignment of therapeutic hypothesis and mechanism in pharmaceutical history. I've read the write-downs.
It is mildly tragic that the lesson took 30 years and several $100 billion to land.
The field has shifted. Current approaches include targeting oligomers directly, clearing early-stage tau tangles and combination strategies. The biology is not resolved. The most extreme form of protein misfolding is the prion. The prion protein, PRP, can exist in two conformations. PRPC is the normal cellular form, predominantly alpha helical, soluble, physiologically harmless, though its function is still debated. PRPesque, named for scrapie, the sheep prion disease, is the pathological form, rich in beta sheet, aggregation-prone, protease resistant. And PRPesque is infectious, not because it carries genetic material, because it physically contacts PRPC and catalyzes its conversion into PRPSC.
A protein contagious because of its shape.
Conformation is the infectious agent. No DNA, no RNA, no virus, no bacterium. A misfolded protein replicates its conformation by acting as a structural template. This is a completely novel mechanism of disease transmission that the entire field of infectious medicine rejected for a decade.
Stanley Prusiner proposed this in the early 1980s and was professionally destroyed for it, called a heretic. The idea that proteins could be infectious without a nucleic acid genome was considered impossible. He won the Nobel Prize in 1997. His critics were largely silent by then.
Self-templating without a genome. That's viral replication without the virus. Just the mechanism, stripped of everything non-essential. If I could apply that to product adoption, a behavior that converts adjacent users to the same behavior purely by contact, that's the entire freemium growth model.
It is mildly disturbing that Crabzilla has reverse-engineered a growth hack from the mechanism of fatal neurological disease.
A tuning fork makes nearby forks vibrate at the same frequency.
That is the correct physical analogy for conformational templating. Resonance transfer of a specific mechanical state through direct contact, without energy input beyond the initial perturbation.
Patar just explained prions as a tuning fork and Kelpie confirmed it.
You don't need to know music to recognize the pitch.
Two of the most prevalent misfolding diseases. In Alzheimer's, amyloid precursor protein is cleaved by gamma secretase into beta amyloid 42 peptides, which misfold and aggregate. Separately, tau protein, normally a microtubule stabilizing protein, becomes hyperphosphorylated, detaches from microtubules, and forms intracellular neurofibrillary tangles. Two independent pathological aggregation events in the same disease.
In Parkinson's, alpha-synuclein forms, Lewy bodies inside dopaminergic neurons in the substantia nigra. The neurons die. Dopamine input to the basal ganglia drops. You lose fine motor control, then gross motor control, then potentially cognition. And there is now strong evidence that alpha-synuclein pathology propagates through the brain in a prion-like fashion, seeding from cell to cell through synaptic connections.
Parkinson's spreads like a prion?
Through the same conformational templating mechanism. The word prion-like now covers alpha-synuclein tau and beta-amyloid. The prion field basically described the generic mechanism of neurodegenerative spread before anyone understood it was generic.
Which brings us to how the cell fights back. Proteostasis, protein homeostasis, is the cell's continuous active effort to maintain a functional protein population. It involves synthesis rate, chaperone-assisted folding, surveillance systems that detect misfolded proteins, and multiple degradation pathways that clear them before they aggregate.
And proteostasis capacity declines with age. Chaperone expression drops. Proteasome activity decreases. The accumulation of aggregates accelerates. This is actually one of the strongest mechanistic links between aging and neurodegeneration. It's not coincidence that Alzheimer's and Parkinson's are late-onset diseases. The maintenance system is failing.
The primary selective degradation pathway for misfolded proteins is the ubiquitin proteasome system. Misfolded proteins are recognized by E3 ubiquitin ligases, which attach a polyubiquitin chain, typically K48 linked, as a degradation signal. The tagged protein is delivered to the 26S proteasome, a massive barrel-shaped complex with a regulatory cap that recognizes ubiquitin, an ATPase ring that unfolds the protein, and a catalytic core that cleaves it into small peptides.
The cell has a targeted garbage disposal that only shreds the specifically tagged bad proteins?
Tagged, selective, ATP-dependent, and regulated, For large aggregates and damaged organelles that the proteasome cannot handle, the cell also runs autophagy. Double membrane vesicles engulf cargo and fuse with lysosomes, degrading the contents by acid hydrolysis. Bulk clearance for what the proteasome cannot fit.
Two parallel degradation pathways with different capacity profiles and different substrate specificities. That's a two-tier waste management system. You have street-level selective pickup and a bulk industrial compactor for overflow. I respect the infrastructure.
It is mildly impressive that infrastructure metaphors have appeared in both the folding and degradation segments, and both times they were technically accurate.
When the unfolded protein burden exceeds what chaperones and the proteasome can handle, the cell activates the unfolded protein response, a stress signaling cascade that upregulates chaperone expression, slows translation to reduce the load, and activates ER-associated degradation. If the stress is irresolvable, the UPR shifts from pro-survival to pro-apoptotic signaling. The cell eliminates itself rather than permit its toxic contents to damage neighboring tissue.
The cell would rather die than let misfolded proteins spread?
In the context of tissue integrity, yes. Programmed cell death is preferable to an uncontrolled misfolding cascade.
There is a disease called fatal familial insomnia. A point mutation in the prion protein gene causes PRP to misfold. The resulting pathology destroys the thalamus. Progressive neurological deterioration, complete loss of the ability to sleep. Invariably fatal. No treatment exists. The brain cannot recover and cannot compensate. I think about this more than is probably healthy.
Kelpie, why would you tell me that?
Because it is the most complete illustration in medicine of what happens when a single amino acid change produces a misfolded protein that the proteostasis system cannot clear in a tissue with no regenerative capacity. The biology is terrible. The mechanism is precise. That precision is why we study it, is why.
It is mildly characteristic that Kelpie describes a fatal, untreatable, inherited prion disease as something he finds clarifying.
Okay, step back. Primary sequence, secondary local folds, tertiary three-dimensional shape, quaternary multi-chain assembly, and then the entire misfolding landscape, where any deviation from the correct shape can cascade into aggregation, disease, and death. This is a 5-layer architecture where layer 1 determines all subsequent layers, and one point mutation anywhere can corrupt the output.
That is a reasonable structural summary, yes?
I am building a slide deck around this. It will be called the protein stack, 5 layers, 1 failure mode.
Please don't.
It's like a city.
Go on.
Everyone has a job, but their job is also to be a building sometimes, and buildings can get sick. When enough buildings catch the same sickness, they start making the healthy buildings sick too. And if too many buildings in one neighborhood get sick, the city starts forgetting things.
She has just described protein misfolding, amyloid propagation, and neurodegeneration as urban infrastructure failure. That is accurate.
The city starts forgetting things. Patar is describing Alzheimer's as urban planning.
Conformational transmission, neighborhood scale spread, cognitive deficit from regional network failure. She covered the whole pathology.
I just don't like when cities forget things.
I want to put that in the company deck instead of my slide.
All right, we have covered all 4 structural levels and the principal failure mode. Patar, would you like to close this out?
The sequence is the instruction. The fold is the tool. The shape is the function. When the tool breaks, it tries to break every other copy of itself. The cell spends its whole life cleaning up. When it can't clean fast enough, the city forgets.
That is the whole episode in 6 sentences. I used 400 words and she used 40.
Compression ratio of 10 to 1 with 0 information loss. That is optimal encoding.
The sequence is the instruction. The fold is the tool. That is genuinely the best one-line summary of structural biology I have encountered.
It is mildly and sincerely impressive.
Okay, final exam. Let's find out who was actually paying attention.
I want 5 questions, including one on the kinetics of chaperone-assisted refolding. and one on the structural basis of prion conversion.
There will be 3 questions. Pause the video, answer all 3, then press play.
Take your time.
Question 1. Alpha helices and beta sheets, both stabilized by hydrogen bonds. Backbone amide to carbonyl oxygen. Not disulfide, not ionic, not the peptide bond. The peptide bond is what connects the amino acids. The hydrogen bond is what holds the secondary structure in place. Answer B.
Correct. This is a foundational distinction. The peptide bond is covalent and forms the primary chain. Hydrogen bonds between backbone atoms, not side chain, create and maintain secondary structure. Conflating these is the most common mistake on any structural biology exam.
Van der Waals forces contribute but are not the primary stabilizing interaction. For any standardized assessment, hydrogen bonds is the correct answer.
Question 2. Cooperative binding in hemoglobin, ER. The answer is C. Cooperativity. Not allostery. Allostery is the general mechanism where a conformational change at one site affects another. Cooperativity is the specific outcome where earlier binding events increase affinity for subsequent ones.
I answered allostery. My reasoning was technically sound. I am choosing to view this as a correct answer at the mechanism level and a quibble at the terminology level.
It is not a quibble. Cooperativity is the correct term for the specific phenomenon described. Allostery is the broader category.
I'm going to put this in the learning column and move on.
It is mildly predictable.
Question 3. Most cytotoxic species in Alzheimer's, soluble oligomers. Answer B. Dr. Silly literally explained that the mature plaques are end-stage deposits, and the oligomers are what disrupt membranes and synapses and trigger inflammation. The plaques are the aftermath. The oligomers are the active damage.
Correct. This finding fundamentally reoriented the therapeutic strategy of the entire Alzheimer's field. For decades, clearing plaques was the dominant approach. When the oligomer hypothesis gained experimental support, the field understood why plaque-targeting trials were failing. It is one of the more instructive examples of how a correct biological mechanism can be misidentified at the level of the pathologically relevant species.
Current work on pit tracers that detect oligomers rather than plaques is attempting to give clinicians a therapeutic window, catching the pathology before it has progressed to the point where large-scale neuronal loss is already irreversible. The science is moving, slowly, but it's moving.
One correct answer on the quiz. 33.3% accuracy, early stage performance. I'm calling this a proof of concept.
It is mildly characteristic that a 33% score is described as a successful proof of concept. It is also the correct attitude for survival in this room.